We have an office here.
Well, if we could all reassemble, those of you who have friends out in the coffee area could go out and make them come in. We can start going. I'd like to welcome Dr. Eberhard Voit, who's come a long way to join us here. And he is earned his master's degree in biology and mathematics, and then a PhD in developmental and theoretical biology from Cologne University, Germany. He's held faculty positions and research positions at the University of Michigan, the Medical University of South Carolina, and at a forest research center in Tasmania, Australia. He's now the chair professor in Georgia Research Alliance, eminent scholar in the Department of Biomedical Engineering at Georgia Tech University. Dr. Voit will share with us his research on the role of systems biology in drug delivery and predictive health. Dr. Voit.
Thank you so very much. I have to admit that I had an invitation last year to come, and I said, what the heck am I supposed to talk about? You know, I do systems biology, talk about nano and tele, and so this year I got another invitation. I said, these people are insistent, and I just do it. If they think it fits, okay, I'll do it. So ignore the abstract in the book, but I will talk about the title that is given there. And let me see. Okay. So I want to talk about systems modeling and systems biology, and since this is sort of an outside topic in this conference, I thought instead of giving a very technical talk, I give an overview of some of the issues that I see that where systems biology could help in predictive medicine and maybe overlap with some of the things that most of you are doing. So I want to talk about the grand challenges of systems biology and then talk about the drug pipeline. And most of you have probably seen the drug pipeline and the different phases of that. And I just want to focus on where systems modeling, biological systems modeling, might play a role in the drug pipeline and maybe help move the screening earlier in the drug pipeline. And then I will talk about pathway modeling and a little bit about predictive health. And let me give you a caveat. So everything will sort of sound like hand waving, but there is some real science behind it, and it will not necessarily show up. And I thought I should say that. And if you're interested in these things, you know, I'll be happy to talk with you or send you papers or whatever. But don't be fooled by looking at pretty pictures. So the grand challenge of computational systems biology is that we have cells, or we have a tissue, or we have an organism. And what we would like to do is convert that into diagrams and with arrows and boxes and maybe different colors and different types of arrows. And we want to take those diagrams and convert them into mathematical equations or systems of equations. And in typical cases, these are differential equations, but not necessarily. There are other things. Then we need to convert that into computer code. And if we have done that, the main thing is we want to come back and gain insights or create some new hypotheses based on the insight that we have learned from the computation analysis. So why do we want to do that? So what are the rewards for solving these grand challenges? Well, of course, the testing would be incomparably faster and cheaper. We can explain root causes of observations. So if you see that phenomenologically you have some problem at the end of the system, you can go back and find out where exactly this problem comes from or what combinations of problems have led ultimately to the observation we make. We would like to make reliable predictions, and clearly, if you have a computational model, you can make hundreds of thousands, millions of predictions in a very, very short period of time, much, much faster than any organismal system or in vitro system. So we can explore whole parameter spaces with these types of models, and as long as the models are, or as soon as the models become reliable, we can make very interesting predictions of what these systems are doing. Then ultimately we want to create novel biological systems, and that's going into synthetic biology and maybe coming a little closer to what you are doing. And in a way we could reduce some animal experiments and ultimately improve medicine. So here's sort of I put this in yesterday after I'd heard the talks, and here are sort of the relationships to what we are doing, what you are doing, the way I see it. So we have some medical examination or methods of telemedicine, and from that we get some biomarker profiles, and here this is a mass spec, but it could be other things like blood pressure or pressure inside the heart or things we heard about yesterday. And that would lead to the identification of a problem and of the affected tissue. And then you could go into location issues, maybe with nanodots, and our department is doing a lot with that, with quantum dots and those things. And you can also go into DNA arrays and look at the genetic predisposition, the genetic makeup, and the expression, changes in expression. Those things all would go into a computational model. This model would allow us to personalize the system to a given patient, to a given person, and then we could customize a viral vector or a nanobot. I don't know much about nanobots. We heard yesterday about the potential of those. And then not just kill the cell that is affected, but maybe correct it in some way. And then everybody would be happy. So here's the drug discovery pipeline very quickly. You know it starts with the discovery phase, and then there's the preclinical development, and the clinical development, and then the postclinical. We have the animal studies, the clinical trials, and so on. And I will not go through this in detail, just say that it takes about 10 to 20 years, and it may cost about $1 billion to come up with a drug that actually makes it to market. And all these steps in the drug pipeline are necessary, and we do not want to cut corners. So the only thing we can really do that would help us in reducing the time and the costs involved with the drug development process is to screen out things earlier than we may screen them out now. So once a new chemical entity, an NCE, goes into clinical phase 1 or 2, it becomes very, very expensive. And if we could, with any sort of modeling or other methods, move the screening out earlier, it would save an enormous amount of effort. So there are different types of models, and here are some that are being used in the pharmaceutical industry right now, and I'm not going to talk about them. So there's screening, there are the quantitative structure-activity relationships, and there are binding predictions. And I will not talk about them because that is sort of going on right now, and I don't need to mention it. There are the PBPK models, that's physiologically based pharmacokinetic models, and they have been in the industry for some while, and they are becoming more interesting and more sophisticated. So essentially what you do is you divvy up the body into compartments, like the blood compartments, brain, lung, and so on. And they are connected, of course, for instance, through the bloodstream. And now you can put a drug into the bloodstream or into the stomach or into the lung and see the kinetics or the dynamics with which these drugs go into different compartments, how long they stay there, whether they come out unharmed in some sense or whether something happens to them. And the different compartments can respond very, very differently. For instance, if you have fatty tissue, it usually keeps lipophilic substances for much longer. If a compound goes into the liver, it is at risk at least of being metabolized, which may be desired or may not be desired. You create breakdown products, which you could follow in the same model. And the beauty of this is you can set up these models in a mouse experiment, and you can validate them. You get the parameters out, for instance, the diffusion rate, the retention time, and other parameters. And then, to some degree, you can scale up these models by saying, well, a mouse weighs 20 grams, we heard, a human is 3,500 times heavier.
3,500 times.
That is 3,500.
Oh, you are.
Sorry. I listened to you. So 3,500 times bigger. If we could look at the volume of the liver or volume of the fatty tissue, then we can make deductions, mathematical deductions, of how long a particular drug would stay in a particular compartment. And these models are very successful, and I think they will become more successful when we learn more about the degradation processes in different organs and kinetics of excretion and so on. Here's something that I was not aware of until last year when I talked with somebody from the pharmaceutical industry. And he said a big problem where we use systems modeling is actually receptor binding. So the situation is that we have a disease process, and the disease process has something to do with ligand binding to a receptor. And very, very many diseases are of that nature. He says we can construct an antibody to that. And when I say to that, there's a question. The question is, should we do it to the receptor? So we can eject the antibody. Antibody binds to the receptor and blocks it from binding the ligand. Or should we generate the antibody for the ligand? And it would bind the ligand, preventing the ligand from binding to the receptor. Now, you cannot, with hand waving, figuring out which one you should do, because it depends on all kinds of characteristics of that system. For instance, how fast and how often receptors are internalized into a cell, how often new ligands are formed, how fast they are degraded or excreted by the kidneys, and so on. So you can set up mathematical models, and it looks more complicated than it actually is. So we have our antibody here, and here's our ligand, here's our receptor. Here's the natural binding between ligand and receptor. It forms a complex that's reversible. The ligand is produced, the ligand is excreted, the receptor is produced, the receptor is internalized or degraded. And then we have the various complexes, and we can measure on animal experiments or in vitro sometimes what the affinities are for the different combinations and for the formation of these complexes. And then we can construct time curves. So this the x-axis is time. Here's the percent of ligand that is left after some treatment. Here is the concentration of this complex 3 here as an example. And you can probably not see the details, and it's not important, but it says, depending on what our initial state is, how many ligands we have, how many receptors we have, and what all these affinity rates are, we can get very, very different answers to this question. In some cases, it's better to develop the antibody against the ligand. In other cases, it's better to generate the antibody as a binding to the receptor. And here, for instance, you see that for small L0s, it goes down and it takes a long time. If L0 is 0, the ligand goes down, it's immediately back up. So that's not a good situation. But if you have L0 equals 100, so that's the ligand concentration at the beginning, you can see that this complex stays in the body for a long time. And that might be a problem, might not be a problem, it depends on the situation. We can do real systems. Here is, for instance, a MAP kinase cascade, and there's a lot of modeling going on with MAP kinase cascades and with crosstalk between cascades. And the beauty of mathematical modeling, of course, is that it's essentially unlimited in size. We can deal with hundreds of components. The hard part is to find the right parameters. Well, we can set up models. Here's another example that we dealt with a long time ago. It has to do with purine metabolism. And so this is sort of one of the types of models we set up. And you can see the letters in the boxes. Those are chemical compounds, metabolites. The heavy arrows are chemical reactions, biochemical reactions, usually enzyme catalyzed, and then the thin arrows are activation and inhibitions. And you can see that this is not a trivial thing to analyze. So if I would ask you what happens if we cut this step here, either because of a mutation or because of a drug, what would happen? You would have a hard time saying what would happen. In fact, it is impossible because it depends on all the numerical combination of this. So what can we do with something like that? Well, one of the diseases associated with purine metabolism is too much uric acid. And for the guys, particularly here, for the old guys, and more particularly, gout is one of the hallmarks of this. So you have too much uric acid, you develop gout, and but your life expectancy goes up for unknown reasons, so you can enjoy it longer. And so we can see where does it come from? Why do we have too much uric acid? Well, we can test hypotheses, can explain it. For instance, if you have superactivity of this first enzyme, then the stuff goes out and most of this comes back here. And then we say, okay, here's another one. If you have this enzyme deficiency, that's very bad. It's called Lesch-Nyhan syndrome. People are retarded. Kids usually die before the age of 2. And it's one of the consequences is uric acid accumulation in blood. Then we can say, how can we intervene with the system? Now, if this model were reliable, which I would not trust it to cure my son with this. I mean, we're not at that point. But we will come to that point. And if this type of model would be reliable, we could now test every possible intervention. And, for instance, we could say, well, what happens if we block uric acid production here? Well, it doesn't take a rocket scientist to figure out what happens. You have less of this uric acid if you block the step. So that's not rocket science. This is much harder. Or we can block it up there, and there's a drug that's working here. And then we can ask for side effects. Now, we can only look at side effects within the system. We cannot look at headaches and constipation, those things. But we can see, for instance, if we go with this mechanism here, with this allopurinol, then the uric acid may go up, but the xanthine, which is the last one here, is likely to increase or to accumulate because it has no way to go. So that might be bad, that might not be so bad. But we can test these types of things, and we could go combinatorically through the whole system and figure out what these systems do. So here's the task of personalized medicine now. The status quo is that medicine is really based on averages, and the averages come either from epidemiology or from animal experiments, usually from combination of the two. And the task for personalized medicine is to progress from average input-output relationships to a deeper understanding of disease processes in individuals. And the challenges, two challenges, are to get data and then to analyze them. And those are our challenges. Your challenges would be, well, how do we deliver a corrective action? And I have not mentioned that here. So the analogy is like engineering, that if you really understand how a machine is put together, you don't have to take every single machine apart. You understand the principles, and even though a clockmaker had never seen a particular clock, he or she would know what the problem is if there's a cog missing or so. So we need to understand the principles behind these types of systems before we can really have targeted corrective actions. So the modeling approaches that can help us in this are sort of various sophistication, let's say. And here's sort of the time axis. We can look at biomarker associations, biomarker networks, and I'll go through that. And then parameter variation in disease models, and then the long-term development of disease. And I want to talk about something new that's called disease simplexes. So with biomarker modeling, in some cases, it's easy. So we have one SNP here in the hemoglobin gene, and we get sickle cell anemia. That is easy to diagnose. It's not an easy disease, but it's easy to diagnose. Things like the Oncotype DX test that looks at 21 genes. So that's a whole combination, and those two have to do with remission of breast cancer. So what we really want to do is go into hierarchical networks of biomarkers. That's where it has to go. So we want to look at disease. We want to find out what biomarkers are, and there are very, very many biomarkers, things that are associated with disease right now. That is not sufficient. So we need to look at some that may be important, maybe not, we don't know. But then we want to look what biomarkers give us some indication that 10 years later you may have a biomarker that is really indicative of the disease. And you can see how this goes. So you would like to look at apparently healthy people and see what their status is, follow their trajectory over time, and see whether the trajectory is going the right direction or the wrong direction. And I'll come back to that in a little bit. And Georgia Tech is working with Emory on a predictive health institute, and that's what they would like to do conceptually. They are starting analyzing lots of biomarkers in healthy people, young people and old people who are apparently healthy, you never know, and then they would follow them for the next 20 years or so, or 50 years, or however long they can afford it, and then develop trajectories that lead from health to disease, metabolic disease, neurodegenerative disease, cardiovascular disease, those things. So the real crux is that we get into dynamic disease modeling, and I showed you the purine metabolism example. That is sort of an example. And now we need to go into the personalization of those types of models. How do we do that? Well, we can develop a physiological model. That's what everybody is doing right now in disease modeling. So you look at what is normal, what are the rate constants, what are the Michaelis-Menten constants, what are fluxes, concentration, and so on. Many people have done that. We are doing that. And you do that by setting up equations, and then you identify parameter values for average healthy individuals. And now you can start personalizing these models by starting with the average model and then adjusting either singly or in combination the parameters for particular individuals. So the assumption is that everything else is sort of normal, at least in the normal ranges. And now you say, well, your cholesterol is high and your blood pressure is high and this one is low and this is so and so. And you want to put that in the model to see what the immediate effect is, what the long-term effect might be, what the effect is without intervention, with intervention. So if we compare that with what's going on right now, so here's the traditional medicine. We have epidemiology, molecular biology, biochemistry, physiology, and so on. Epidemiology gives us sort of hypothesized risk factor disease associations. You get lung cancer if you smoke. Your relative risk is whatever it is, 4 times. It goes into physiological mechanisms. The physiological mechanisms usually come from animal experiments. From these mechanisms and from animal experiments, we get process parameters. That comes all from traditional biology, targeted biology. These things, physiological mechanism and process parameters and drug treatments go into clinical trials, and you get an average treatment. So you weigh so and so much, here's the dose that you should get. If we look at systems biology, there's experimental systems biology. I'm talking about microarrays, high throughput screening, proteomics, metabolic profiles, those things. It actually adds to these 2 boxes here. And if we look at computational systems biology, we can set up models of averaged people, of physiological models. We can perturb them and do simulations of diseases in the way that I just showed you. We can also look at treatments of these diseases. We can go and almost exhaustively screen for target sites, intervention sites. That's all done with numerical solution. Now, these models will allow us to do personalized simulations, as I indicated a minute ago. And so here we adjust the parameters to match a particular person. So we go into personalized health, personalized treatments, risk profiles, and so on, and then have suggested prevention. So if we look at those biomarkers, they have all normal ranges. If we put them in 2 dimensions, they seem to have little rectangles. Now we know that combinations are often not very good for you, so these boxes actually have their corners cut off, and so you get a shape. If you do that in many dimensions, you get something like a simplex. So it's like a diamond that's cut off. So if you're inside this diamond in the simplex, you are healthy. If you're outside, you are not necessarily. And we can compute, at least in principle, these simplexes from the model. So ideally, we would have a full parameter space and a full biomarker space. We would have a health and disease simplex. They are probably nested like that. And we would like something like the PSA, where we say, well, if it's higher than 4, you have a problem. If it's lower than 4, you're okay. So it looks like a plane cutting through the simplex. And you say, well, if you're on this side of the x-axis, you're okay. If you're here, you're not. Now you can see that is sort of treacherous, because depending where the other components are. So this would be healthy. This person would be healthy. A person here you don't necessarily know. A person here would be diseased because he is outside this health simplex. Now, as I said, ideally we are learning how to compute these simplexes from our computational models, and it's a matter of parameters by and large. So looking at one biomarker is clearly insufficient. One needs to do more in combinations. So if we do it in 2 dimensions, we can look at trajectories. Here's health. Here's something like pre-morbid conditions. Here is something like treatable disease, and outside that is bad. So that's pre-morbidity. Here are 3 people. The first one gets a little sniffles, not too bad, comes back and is healthy again. Here's somebody who has problems, gets a pacemaker, is back. Here's a person that is not getting the right treatment, and boom. So that's a simplified version of what can happen to you. Now, we can, in principle, if we had computational models that are reliable, we could compute these trajectories along the life of a person, and at each point we could say, well, you're deviating to this side or to that side. You are not at risk of Alzheimer's, but you are at risk of metabolic disease, something like that. So in summary, modeling will not replace anything in biology or medicine, but it will complement it. So there's experimental systems biology, but there's the traditional biology, and modeling complements that. There are quite a few technical challenges. At the top of the list, at least for us, is parameter estimation. So to make the model match the actual reality. Systems modeling can play roles at several points of the drug discovery pipeline, I'm sure, and in predictive health. And if we want to go from an average metabolic or genetic or physiological model to a personalized model, I think we need to go into these models that allow us to check combinations of deviations. Physicians do that intuitively. Eventually it becomes too complicated, and these types of models have the potential to help at least. So we have had good funding from a variety of sources, and this is my crew. It's about 1.5 years ago, and there's information if you're interested, and I'll be happy to take a couple questions.
Thank you very much. Questions? Please.
In terms of translating the model that you have you plan to use in predictive medicine to commercial application, is a model something that someone can own and sell? I'm told that the intellectual property gets quite tricky, because you can always add or subtract something from the model and have a new model that you then don't own. Can you comment on that?
Yeah, so the answer was easy about 20 years ago, and it says you cannot own mathematical constructs or theorems or proofs or anything mathematical. You cannot own it. And then people started saying, yeah, we're not selling the model, we are selling the algorithm and some machine that implements that algorithm. Yeah, that was a dirty trick. And now it's really a big gray zone. And I don't think anybody really understands how that works. Now, there are companies out there like Entelos in California that sell models to, for instance, Johnson & Johnson. And they really go through the literature and put everything they can find, put into those models. And they have one for pertussis, and they have a couple others, I forgot what they're about. And they're very complicated models, and they will never divulge what their exact specification of these models are. That's sort of their trade secret. But they do sell them to Johnson & Johnson. So I would imagine that something like that would be happening rather than, patent production or something.
Professor Barinholtz, please.
I think the only way is really trade secret. There is no other way, and go for specific things to do. And you can have a patent on a specific application, but you always have to keep. But that's not what I want to comment. I think we use a lot computational not biology, but computational approaches to more and more things. And there are few problems with them. One, you need a lot of data. Otherwise, it's meaningless. So if you don't have a lot of data, you can use other type of approaches like decision trees and something like that, other ways, like we're using Weka a lot and things like that. But still, I remember when I started with this, I have a very simple problem I want to solve. And actually, think that I knew the solution. So I went to a theoretical computational biologist, and I said, I want you to figure out how many molecules of water are attached per polyethylene glycol of this size. Okay, very trivial thing. So I knew the number. We have it by two different methods. I came to the guy. I gave him the calculation. He came after first time, and it was no resemblance to the real life at all. So I said, no, sorry. He tried again. At the third time, he succeeded to do it. But once he succeeded, now we use his model, and it works every time. So I think he put enough information in it. He asked me a lot of questions. So the problem with these things that you have to find a way to build the model that it will be a robust model and not something that you will have so many false positive and negative in it that it has no meaning, like many of the models are. And that's what bothers me in this, that we become, in a way, dependent on this model. The relationship is very, very strange. The model people are dependent on the practical people, but then you have the opposite relationship. Now, say someone came to you, and even the FDA will say, there is a model, but your data doesn't agree with the model. So you have a problem, or the model have a problem? I think most of the cases, the model has a problem, because they don't have enough information to deal with it, not because it's wrong, but because they still not ready for it. So I think that's really a very, very crucial point that I'm looking for answer to you, being a layman in the modeling, but really using it quite a lot.
Okay, I could talk for about an hour and a half about this answer, which I will not do. Rest assured. So, two short answers. One, in comparison to biology and biochemistry, modeling is very, very young. If you look at the first models that had anything to do with biology, it's not really all that long ago. I still worked with, solving 3 differential equations on a mainframe computer at the University of Michigan, not at Podunk. I mean, this was a good, university, and it's not that long ago. So we are way behind. The expectations are sky high. We are trying to hit the moon with a slingshot, and we're not going to hit it anytime soon. Having said that, if you look at the analogy with physics and engineering, we are able to predict how to send a little machine slingshotting around Jupiter, ending up at Mars, dropping off a little other machine that's running around as predicted and send it photos back. That's where we will come in the long-term future. And if you say most models are wrong, yes, I agree, because we don't have enough information. But I think eventually we will not have a choice anymore. It's easy. If this group and a few physicians got together, you could take 10,000 biomarkers easily of any person in this room. Now, who can evaluate 10,000 biomarkers and you take them, let's say, every week. Who can evaluate that over time? It's not possible anymore. So the models need to become better, but we are still in the slingshot phase, and they will become better. That's our job security.
Well, if I just add my own experiences with this, when we were doing a lot of field work looking at hepatitis B virus and clinical studies and biological studies on the virus, and we wanted to get some kind of overall picture of the dynamics of epidemics and pathology and the consequences in the general population for a very widespread infection. And at that time, I was visiting the institute, the Santa Fe Institute, where they kind of specialized in chaos at the time. And I would show them all this stuff, and the first thing they'd say was, well, which variables can you get rid of? And my reaction was, wait a minute, I've just spent all this time collecting all the variables. I don't want to get rid of any of them. That was an impasse. But I think your prediction, starting off from, well, physics of being able to land a satellite on Titan after sort of 8 years sitting in Cassini is nothing short of remarkable. But biological systems aren't, as I needn't tell you or anybody else here, are not the same. But it's very encouraging, the continued approach.
Yeah, I think if you look at the number of people who actually have devoted a lot of effort to modeling in the past, which is a very, very small number in comparison to biochemists and molecular biologists, and you look at the rate of progress, it is encouraging.
Yes.
But it will take a long time before we're there.